And what I think is hopeful, I just wanted to give you a flavor that we're beginning to have access now to the kinds of genetic tools which should enable us to do that kind of analysis. And I'll describe very briefly this system for mycobacteriophages, but I'd like to think that this is generally applicable to phages of other hosts as well. It is simply a technology that enables you to make a phage mutant or a phage derivative, to add a tag onto a particular open reading frame. There's lots of things we can do with this technology. And it's based upon the following idea. We've constructed a strain of the host, which is referred to as a recombinating strain. It's a bacterial strain where we've actually identified phage proteins that promote high levels of recombination. And so I'm not going to go into the details. I just wanted to give an overview of the technology. If we take this strain, which is highly recombinogenic, we can do the following trick. We take phage DNA. Here's the phage DNA of your choice. And let's imagine that what we're interested in is finding out what gene 55 does. So we want to make a mutant that's lost gene 55. We simply take our phage DNA. We make a small DNA substrate which targets the deletion of gene 55. Got DNA to the left, which is gene 54. DNA to the right, which is gene 56. But this can be any deletion or any change that you want to make. You take this DNA and that DNA, you introduce them into this recombinogenic strain by electroporation, and then we plate out for plaques. In this case, each of the plaques has arisen from an individual cell which has taken up the DNA. What's key about this process is that those cells, not all the cells take up DNA, 0.1% maybe. The ones that don't take up DNA, we don't have to worry about because they don't give us plaques. The ones that do take up DNA frequently, at a very high frequency, take up both pieces of DNA. And this frequency of recombination in this strain that we've engineered is sufficiently efficient that all you have to do is to pick 18 plaques. You just pick 18 random plaques and do PCR analysis, and 10 to 40% of those plaques are mixed containing both mutant and wild type. They're mixed plaques, but all you do is do what the world did. Pick a plaque of the mixed plaque, dilute it out, so you've now got individual plaques. So now you're separating the wild type from the mutant from the population. Pick another 12 or 18 plaques, and you can readily identify a clean deletion derivative of that mutant. And now that you've constructed the mutant, you can ask what its phenotype is, what its properties are. This can be done in a conditional way to knock out essential genes. You can add his tags or strep tags. You can make point mutations, make nonsense mutations, and it can be done relatively simple with one electroporation and two rather simple rounds of PCR. And so we're rather enthusiastic about this. This is a technology that we think can be applied to probably any mycobacteriophage, although we have not tried everything yet. But I'd like to believe that this is not restricted just to mycobacteriophages and mycobacteria. If you can develop these highly recombinogenic strains with other bacterial hosts, this provides a general strategy for constructing mutants and mutant derivatives that hopefully will finally provide the relatively simple high throughput techniques that will open up the prospect of rational phage genetics for all of the genomes that we would like to characterize. So I'm going to just conclude briefly here by saying that I've told you that phage genomes are genetically diverse, but there are related groups of genomes that we can see by nucleotide sequence comparisons. We talked about the mosaic architectures and that these genomes have lots of novel genes or genes that we haven't seen before, as well as unexpected genes, surprising genes, that yes, we don't know what the function of many of the genes are, especially the non-structural genes, but that we're hopeful that the new technologies will provide the kinds of approaches that we need and the techniques so we can address that question. Let me finish by just saying thank you to a few folks, especially Marisa Padula and Debbie Jacobs-Serra, who have coordinated our HHMI-funded phage hunting programs in Pittsburgh. Many of the genomes that I've shown you in the latter part of the talk were isolated by students in the lab, high school students and undergraduate students, and sequenced and annotated by them. That certainly would not have happened without the rather miraculous skills of first Marisa and then Debbie. The genomic work that was done in Pittsburgh, I have to acknowledge my collaborators Roger Hendricks and Jeffrey Lawrence, Craig Peebles, several others, including these that have assisted in the sequence analysis, Steve Creasorn for his development of Famarator, David Hanauer for some educational components, and my collaborator, Bill Jacobs, who is at Albert Einstein College of Medicine. And we have to thank Bill for using bacteriophages right back in the late 1980s to develop genetic systems for TB and the mycobacteria, and definitely helped to spur all of our interests